When we think of forensic science, we often picture crime scene investigators dusting for fingerprints or extracting DNA from a sample of blood or tissue. But in recent years, a new kind of genetic sleuthing has emerged that doesn't require a drop of blood or even the capture of an animal. It's called environmental DNA, and it's revolutionizing how scientists track some of the world's rarest, most elusive, and most beautiful creatures. Hello, and welcome to this channel. Today we're re-diving into the world of wildlife genomics and environmental DNA and how it can help us understand and protect some of our planet as most elusive creatures without ever having to capture them. Wildlife genomics is an interdisciplinary field that combines elements of ecology, genetics, and conservation biology. It involves the large-scale study of animals' DNA and their natural environment with the aim of understanding the genetic makeup and evolutionary history of populations and species. This can provide crucial information for conservation efforts, helping scientists to identify and protect endangered species, monitor populations over time, and understand how species interact with each other and their environments. One of the key tools used in wildlife genomics is environmental DNA, or eDNA, which is exactly what it sounds like. It asks the DNA found in the environment that can tell us a lot about the species living there. The idea behind eDNA is pretty simple. When an animal moves through its environment, it leaves behind traces of itself, such as skin cells, feathers, hair, or urine. These leave behind genetic material in the form of DNA. By collecting and analyzing this DNA, scientists can identify the species present in an area without having to actually capture them. This might sound like a far-fetched idea, but it's surprisingly effective. And it's made possible by advances in DNA sequencing and bioinformatics, which allow scientists to analyze vast amounts of genetic data quickly and accurately. There are three main ways that eDNA is being used to study wildlife: to detect the presence of a species, to track the movement of a species, and to study the genetic diversity of a population. Let's start with species detection. This is probably the most straightforward use of eDNA, and it's already being used to great effect in a variety of ecosystems. For example, in the Amazon rainforest, researchers have used eDNA to identify hundreds of tree frog species from samples of water collected in the forest canopy. In Australia, scientists have used eDNA to detect the presence of the platypus, a notoriously elusive creature that's been hunted to near extinction. And in Yellowstone National Park, researchers have used eDNA to monitor populations of gray wolves, which are an important indicator species for the health of the entire ecosystem. In all of these cases, eDNA has been able to detect the presence of these species in areas where they were either rare, elusive, or difficult to study using traditional methods. It's also been able to do so with a high degree of accuracy thanks to advances in DNA sequencing and bioinformatics. But eDNA's ability to detect species isn't limited to just animals. It can be used to detect plants, fungi, and microbes as well. For example, in the wine industry, eDNA is being used to detect the presence of harmful bacteria and fungi in grapes and wine, which can help producers to ensure the quality and safety of their products. The next way that eDNA is being used to study wildlife is to track the movement of a species. This can provide valuable information for conservation efforts, such as identifying migration patterns or tracking the spread of an invasive species. For example, in Florida, researchers have used eDNA to track the movement of the Florida black bear, a threatened species that faces a number of conservation challenges. By analyzing eDNA samples collected from different locations, scientists have been able to map the bear's movement patterns and identify areas that are important for their survival. Similarly, in Hawaii, researchers have used eDNA to track the spread of a highly invasive and destructive rodent known as the small Indian mongoose. By identifying the areas where the mongoose is present, conservationists can focus their efforts on controlling the spread of this invasive species and protecting native wildlife. Another exciting application of eDNA for tracking species movement is in the field of fisheries management. By analyzing eDNA samples collected from fish farms and hatcheries, scientists can identify the fish species present and assess the risk of invasive species. This can help managers to make informed decisions about stocking and harvesting fish populations, ensuring that they are both sustainable and free from invasive species. The third way that eDNA is being used to study wildlife is to study the genetic diversity of a population. This can provide insights into a species' evolutionary history and help scientists to identify populations that are at risk of extinction. For example, in the Serengeti National Park in Tanzania, researchers have used eDNA to study the genetic diversity of elephants. By analyzing the elephant DNA shed into the environment, scientists have been able to estimate the size of the elephant population and identify individuals that are related to each other. This information can help conservationists to develop effective strategies for protecting the elephants in the park. Similarly, in the Amazon rainforest, researchers have used eDNA to study the genetic diversity of tree frogs. By analyzing the frogs' DNA shed into streams, scientists have been able to identify different species and populations of tree frogs and assess their genetic diversity. This information can help conservationists to protect the genetic diversity of these frogs and ensure that they are able to adapt to changes in their environment over time. So those are the three main ways that eDNA is being used to study wildlife. It is a powerful tool that is already being used in a variety of ecosystems to study some of the world's most elusive and beautiful creatures. Now let's talk about the benefits of using eDNA over traditional methods of studying wildlife. The first benefit is that eDNA can be used to study species that are elusive or difficult to capture using traditional methods, such as the Florida black bear or the platypus. This means that scientists can gather data on these species without disturbing them or their habitat, which is especially important for endangered or threatened species. The second benefit of using eDNA is that it can be used to study species that are rare or have a low population density. For example, in Yellowstone National Park, gray wolves were reintroduced in the 1990s after having been hunted to extinction in the park. By using eDNA to monitor wolf populations, conservationists can get an accurate count of the wolves in the park without having to trap and collar them. Another benefit of eDNA is that it can be used to study species that are active at night or during other non-standard hours. Many animals are crepuscular, meaning they are most active at dawn and dusk. This makes it difficult to study them using traditional methods that require researchers to be out in the field during standard daylight hours. But eDNA capture the genetic material shed by these animals when they are active, allowing researchers to study them even if they are nocturnal. The fourth benefit of eDNA is that it can be used to study multiple species simultaneously. This is because eDNA samples contain DNA from multiple sources, which means that scientists can analyze a single sample to identify the species present. This is especially useful in ecosystems that are rich in biodiversity, where there may be hundreds or even thousands of different species present. Finally, the fifth benefit of eDNA is its non-invasive nature. Because eDNA require researchers to capture or handle animals, it is a non-invasive method of studying wildlife. This is important for protecting both the animals and the researchers. Some animals may be dangerous to humans, while others may be threatened or endangered. eDNA allows researchers to study these animals without putting them at risk. Thanks to the incredible advances in DNA sequencing and bioinformatics, eDNA has become a powerful tool for wildlife genomics and conservation biology. By using eDNA, scientists can study elusive, endangered, nocturnal, or rare species without disturbing them or their habitat. This allows researchers to gather valuable information about these species and their ecosystems, which can help to protect them and ensure their survival for generations to come. I hope you enjoyed learning more about eDNA and how it's being used to study wildlife. If you did, please be sure to like and subscribe so you never miss an episode. Thanks for watching, and I'll see you next time.